Hello, everybody. Welcome to CellTalk. Talking about CellTalk, it is a platform where we enlighten you with knowledge in regard to health sciences and covering topics on various where he will be shedding light on domains of drug repurposing for the disease glioblastoma multiforme. I'm your moderator, Dr. Chiku Cherian, PhD in bioinformatics. Our speaker, Souvik, is a scientist and content writer at Renix. He writes scientific content for blogs, updates, review papers, and clinical white papers. His other activities include communicating with scientists and delivering scientific content. He has completed his PhD specialization in immunology and clinical biology. His research is host-pathogen interactions in diseases through immunologic profiling. Before joining Renix, he worked at Basuda Center for Interdisciplinary Sciences. Now, these are the instructions to be followed for the webinar. Questions by the audience will be answered after the session by Dr. Souvik, where you can type your questions in the chat box. The questions pertaining to the research will be selected, and the person can unmute and ask the question to Dr. Souvik, where we would be happy to address them. Over to you, Dr. Souvik.
Thank you very much for this enlightening speech. And let's begin this webinar. We have, decided to select glioblastoma multiforme as our disease of priority for this session. And as Chiku has rightly mentioned, the theme for today is repurposed drug in glioblastoma multiforme. So I have chosen one paper whose title goes like this. It evaluates the effect of metformin. As we all know, metformin is a very common oral drug that is used in diabetes. So we will going to go into the journey to investigate this effect of the metformin in vitro culture and how it can be used as an add-on therapy against glioblastoma. So before going into the details of the investigations and its results, I would like to give a brief on the journal that I have selected for. It's a Cancers journal with an impact factor of 6.6, which is quite a very good impact factor. And more importantly, it has been recently published on March 2022. So what is metformin? Metformin, as we all know, is a very common oral drug, which is used specifically for the treatment of type 2 diabetes mellitus. And this has a profound effect as a repurposed drug in many diseases like polycystic ovary syndrome, pancreatic cancer, as well as hepatocellular carcinoma. Along with this, there are many clinical studies that is going on with metformin in ovary cancers and as well as in colorectal cancers. So let me give a brief about what is and to which extent GBM is prevailing in our society and why we should come up with an alternative or a kind of repurposed regime of drug for this disease. GBM or glioblastoma multiforme is a malignant cancer of the brain and is an aggressive grade 4 astrocytoma. Means it is the cancer of astrocytes where during the propagation stage, it often goes to malignancy and metastasis. And it has been the cancer patients who are suffering from this lethal disease, they are having a very, very small lifespan after being diagnosed with this disease. They have around a median survival of 14 months. So you can see the aggressiveness and the morbidity as well as the mortality issues related to this cancer. And recently, I can say recently within last 8 to 9 years down the line, there are many investigations that is going on to search out for an alternative treatment regime. Let me say, what is the standard treatment regime for glioblastoma multiforme? It is till now it is based on a conventional therapeutic approach involving surgical resection followed by an adjuvant therapy with temozolomide as well as radiation therapy, which are both very toxic in nature. So in light of these challenges, the investigators and researchers all over the world are searching for some alternative treatment regime. And very interestingly, in many past researches and investigations, they have found that the diabetic patients who are also comorbid with this lethal disease have a very profound effect of metformin on their system, and it can reduce the tumor cell propagation. So this interesting lead results in further investigation of this metformin drug in the research of glioblastoma. So as I have told you, glioblastoma is one of the most deadliest brain cancers, and there are still some challenges in this domain, in this brain cancer. The first one is the recurrences in GBM tumors, and it is always happens like, if a patient got this disease, it is supposed to come back again, even if after surgical procedures. Another challenges for the therapeutics of GBM is the drug resistance. People often got resistant to temozolomide, which is used in excess in order to, if the dose, there are doses of temozolomide that are equally lethal to both normal cells as well as cancer cells. So sometimes the cancer cells are always mutated, always changing its nature. So there happens in many patients that even temozolomide in high doses are not responsive to the patients. And the main reason for that is the mutational instability in the clonal propagation of the cancer cells. So keeping in mind of these three main challenges, let's see what the investigators are trying to find out. So they have designed three objectives in order to study the glioblastoma endothelial cells in culture in vitro as well as in vivo. They have tried to see the efficacy of metformin as an add-on treatment to GBM, along with the normal or the conventional TMZ plus radiotherapy. The second objective they have tried to address is the how much the metformin is involved in bringing the inhibition of angiogenesis. As we all know, angiogenesis is another very pivotal kind of event that occurs during metastasis of cancers. That is the formation of new blood capillary vessels, which facilitate the cancerous cell to spread to the different parts of the body. And likewise, they also try to elucidate the molecular pathway involved in bringing the effect of metformin in glioblastoma endothelial cells. Why they picked the glioblastoma endothelial cells? Because these are the key cells which modulate the formation of angiogenesis and the spread of cancer cells. So let's come to the operational point of view of this investigation. What they have done, they have cultured the GBM cells in vitro from tissue biopsy samples from 10 patients who are already diagnosed with GBM. They have cultured it in the Dalbeco's modified eagle media. That is a media that is used regularly in practice for culturing cancerous cells. After the culture for few phases, subculturing and culturing of the total GBM endothelial cells, there are given the treatment metformin as well as in combination of metformin with the TMZ. So there are 3 sets of experimental experiments that was going on that they have done. One, they have treated the GBM cells with metformin alone. Then they have tried out with the combination therapy of TMZ with metformin, and they have kept another one as without any treatment that has been treated as a negative control. So what they studied the how much after giving this kind of treatment, both the combinational therapy and the monotherapy, how much the the glioblastoma endothelial cells are proliferating and migrating. So they have tried out different kinds of assays to enumerate this process. They have tried to assess the viability of GECs by live or dead assay as well as via MTT assay. So these are two mostly used assays in to sort out different kinds of cells, whether they are viable or not in culture. So after treatment, they try to see whether how many cells they have propagated and how many cells they have it got the right treatment from the the drug and got into the apoptosis. That I will come next. What is apoptosis? Actually, when there is a systematic programmed killing of cells in the body, that is called apoptosis. So if it is the measurement of the efficacy of the treatment, if in the culture during with the help of this kind of treatment, if the cells are going to get into a apoptotic stage, that means the treatment is going good. So we using the caspase 3 and 7 activity assay as well as annexin 5 assay, they have tried to measure the extent of apoptosis in the cell culture. Finally, they have come up with another assay called tube-like structure formation and migration assay to see the spread or the angiogenesis of this kind of the modulated GECs or the glioblastoma endothelial cells in vitro or in culture media. And ultimately they tried to go through different kinds of mRNA transcriptomic approaches like RT-PCR to find the expression of the cells that may lead to the finding of the molecular mechanism behind the arrest of such kind of cancerous cell propagation. So let me directly come to the results part. What they have obtained out of these 5 to 6 assays. Using the MTT assay, they showed that the effect of metformin as well as the combinational therapy with metformin and TMZ brought around an induced apoptosis of the cancerous glioblastoma endothelial cells. As you can see, there are these elongated cells are mainly the GECs or the endothelial cells. So when they have been treated with 20 millimolar concentration of metformin and the combinational approaches with TMZ and metformin of that same concentration, they have seen that with respect to the negative control or the basal media culture, there has been enhanced reduction of the endothelial cells. Further, when they have increased the dose, they found that there are even further killing of the apoptosis of the endothelial cells. So in this way, they tried to see whether the combination therapy is working fine or monotherapy is working fine. So in both the cases, they found that there are substantial reduction of the endothelial cells in culture. Of course, they didn't go for 24 hours culture. They went for around 48 to 72 hours of culture. Then next day, what they did is they have used the fluorescence tracking method to use the live and dead assay in order to see the effect of metformin as well as the combinational therapy. They obtain similar results with this. As you can see in the basal state, there are a huge confluence of cells when it has been treated with metformin of 20 millimolar concentration, it has reduced, and further it has reduced to a significant amount with the higher dose. So as the dose will go higher, it will see there will be a significant reduction of the GECs. Then they performed the tube-like formation assay, in which they tried to see whether upon treatment with TMZ, metformin, as well as with the combinational therapy, is there any reduction of the capillary formation. And these are all microscopic finding. They have put it under the microscope in order to see how much elongated or what is the number of reduction of these tube formations. So they found that with the combinational therapy, they got the best results. And subsequently, they also used a migration assay, which is another very common assay that is used in vitro to see the effect of different therapeutic regime. So similarly, as you can see in the basal media, there has been a huge migration of cells from both the sides. But while after the administration or the treatment with metformin, the similar cells are they're less migrated from this zone. These are the two zones. This is the contact kind of part between the two zones. So with TMZ also there are equal same kind of results with the with respect to the negative control. But while using the combinational therapy of TMZ with metformin, they have seen a significant reduction of the migration of the cells. So all these experiment pointed towards one thing, that is metformin monotherapy or metformin along with the TMZ is showing a kind of arresting kind of the drugs are actually showing an arrest in the propagation of the cancerous cells. So in brief, what we got as a summary of the results is that as most of the patients, the 10 patients, the number is very small, but however, most of the patients have a median age of around 60 years, and they are IDH gene wild type glioblastoma patients. And with the MTT and live dead assay, they showed the most significant effect of metformin at the concentration of 20 millimolar. The third findings, third result summary is the efficacy of metformin is indeed induced apoptosis of these glioblastoma endothelial cells, both with the combinational therapy as well as with the monotherapy. Likewise, the tube-like formation and migration assays also supports the same. They also say that it has a reduced angiogenesis of the GECs or the glioblastoma endothelial cells in both migration of the cells as well as the tube or the capillary formation tendency of the GECs. So ultimately, they also tried to see the transcriptomes. Transcript means the mRNA or messenger RNA on the genes that is encoding the enzymes. And they found out that with respect to the negative control and after treatment, there has been a shift in the upregulation of the genes, especially encoding for the ceramide biosynthesis. That is a player which is involved in a sphingolipid rheostat pathway. So in discussions, we would try to go through the main points that have come up with the research and the investigations on metformin. That metformin can be given as an add-on therapy along with the temozolomide or the standard therapy. There are side effects of TMZ plus metformin, but these are found to be safe and tolerable in the small percentage of GBM cells or GBM individuals. And obviously further investigation of this combinational chemotherapeutic regime needs to be tried in a large cohort-based studies. It should not confine to around 10 or 20 individuals. And obviously the molecular pathway, as I have told you, as a sphingolipid rheostat pathway, it should be investigated down with other targets and other dosage regime of both metformin and any other, derivatives of metformin. So, so these are all in vitro level studies, and preclinical studies are yet to be done in this direction. So what are the impact of this research? What we can get from this research is that we can say that metformin can be obviously used as an add-on therapy with temozolomide in inhibiting the proliferation and expansion of the glioblastoma cells. And this metformin can be repurposed for GBM therapy in future in both diabetic as well as non-diabetic patients. As in case of diabetic patients, it occurs like a serendipity. As you know, they have been given metformin, and they have also been observed that they are having a good prognosis. So in this way, if we can scientifically, we can have a logical turnover with the metformin with temozolomide, and if you can see it in a clinical trials that it can be administered to both diabetic and non-diabetic patients, then there can be some improvement in the therapeutics of GBM or glioblastoma. So what's the third point? Third point is that metformin can be totally safe and tolerable drug. As you all know, it's a very common oral drug. It's an anti-hyperglycemic drug that is used for type 2 diabetes mellitus, and it is also been used in other diseases like polycystic ovary syndrome and gestational diabetes. So it has already been repurposed in many other diseases. So all in all, metformin can be useful in improving the prognosis and clinical outcome of the patients. So this is our takeaway message from this research, is that we can use metformin as an add-on therapy along with the TMZ in order to see it on the effect of metastasizing GBM individuals or malignant GBM patients. So thank you. Thanks a lot for the patient hearing. And I would like to now ask for different questions to come.
Thank you so much, Souvik, for that enlightening session. We really got to learn a lot. So some questions from the audience is, Dr. Vijay would like to ask a question. So Dr. Vijay, you can go ahead.
Nice presentation, Souvik. My question is, what target or pathway metformin seems to be working against in the GBM cells?
Yeah, as I have just pointed out that sphingolipid rheostat is the molecular pathway that it is targeting. Actually, the glucose mainly what happens is that in the diabetes mellitus there is an insulin dependent kind of condition that prevails. So the insulin is not able to absorb the glucose, is not able to facilitate in the absorption of the glucose molecule from the blood. That's why there are increased level of glucose that is found in the diabetic patients. So these are the lipid binding molecules. Sphingolipids are mainly lipid molecules which helps in the absorption of the glucose. So metformin usually works in that way. So when metformin is given to the GBM patients, it is taking out the glucose out of the flux so that the GBM cells can't utilize that glucose as a part of the energy. So
it is starvation.
It's kind of starvation of
yes,
the cells. So
okay,
it is all linked with the the hormonal signaling also.
Thank you, Dr. Souvik. Next, Dr. Nupur would like to ask a question.
Yeah, Nupur.
So, as per I understand, MET is affecting the metabolic reprogramming of the cancer cells.
Yes.
So what will be effect for the non-diabetic patients, because they actually don't need MET? Because
as this is a very good question. For non-diabetic patients, the thing is that for non-diabetic GBM patients, I mean the glioblastoma patients, there are the instances that the cancerous cells used to come into the bloodstream and all. So here, as an add-on therapy, the metformin can be used as an add-on therapy to, lead to the starvation of the glucose over there, so that there can be an early arrest of the glioblastoma cells in those patients. And it has got some results in past. It is not only the in vitro cases. Metformin has been used in colorectal cancer, pancreatic cancer, in the patients, and it has gone through the clinical trials, and they have observed that there has been a significant inhibition and the arrest of the glioblastoma cells, or not the glioblastoma, the cancerous cells. So I think the GBM cells will get arrested in the non-diabetic patients also.
Okay, or even if it does not, it may be used for only diabetic patients. That is also useful.
Yes.
At least
even for preventive or prophylactic cases, it can be used.
Yes.
It can be used.
Yeah, at least a group of patients will be benefited.
Yeah, but the dose optimization needs to be done.
Oh, yeah.
At what dose it needs to be taken and out, because it also has a side effect. Metformin also has side effects.
Okay, at present the result shows it is effective of 20. Result.
Yes, around 20 millimolar of concentration is enough to reduce the glioblastoma cells.
And 50 is too good.
Yes.
Okay, thank you, Shobhu.
Yeah. So any more questions?
So far there is no questions. So anybody else, we'll give some time to ask some questions. Anybody else want to go ahead, can type your questions in the chat box. So I see no further questions coming. So thank you so much, Dr. Sauvik, for that very enlightening session. We could learn a lot about glioblastoma and how metformin and TMZ can work against it. So it really was, we could take a lot of messages home. So I hope to see you all in the next session. And thank you all so much.
Thank you all.